As you know, our gastrointestinal tract is definitely not sterile. From birth, we accumulate a lot of microbes, bacteria, viruses that constitute our microbiome, and in fact, that is very important. And most of the pathogenic organisms, or most of the microbial organisms that we encounter are perfectly harmless or even beneficial, and then some are pathogenic. When they are pathogenic, they often lead to gastroenteritis, they lead to diarrhea, or they lead to long conversations with the big white telephone, so vomiting. And indeed, la diarrea is muy peligrosa in some instances, and definitely when children are either small, undernourished, or in impoverished conditions, it can be very dangerous, and it can lead to dehydration. That is something that you know very well. Rotaviruses are the most important cause of infant gastroenteritis. It took till 1973 before Ruth Bishop looked into the fecal material and the gut samples of children with diarrhea. And what she found was, under the electron microscope with collaborators, this quite typical viral organisms, the rotavirus. They call it rotavirus because if you have a little bit of fantasy, the periphery of the viral particle looks a little bit like the spokes in a wheel, and henceforth the name rota, in Latin, wheel, for this organism. Rotaviruses are extremely democratic viruses in that it doesn't matter if you live in industrialized countries or in developing countries. When you have gastroenteritis, about 40% of the gastroenteritis cases will be caused by rotaviruses. The only difference between, or the biggest difference between industrialized countries and developing countries are the E. coli diarrheas, which are more prevalent in the developing countries. The reason why rotaviruses can do their nasty work is because they destroy the villi that line the surfaces in the gastrointestinal tract. So the big columns of cells, they are destroyed, and when they are destroyed, they become a lot smaller, they become stunted, and of course that leads to a smaller surface and a smaller possibility to reabsorb or to absorb nutrients and water. And in fact, rotaviruses work via many mechanisms to either reduce the absorptive surface, functionally impair the absorption by many ways, of course kill the cells or damage the cells, which does not allow them to have their function. The biggest contributor to the enterotoxigenic effects is the NSP4, the non-structural protein 4 of the rotaviruses, and so on. A couple of days after and it only takes 3-4 days, and it probably depends on the initial viral inoculum exactly how long it takes. After the infection, the patient starts the shedding of the rotaviruses even a day before the symptoms become clear. The symptoms often start with fever and vomiting rather than with diarrhea, which is for some parents counterintuitive when you talk about gastroenteritis. And then finally the diarrhea starts, and then depending on the level of dehydration and the level that the parents are worried, a child might be hospitalized for that. Or if it's not hospitalized, it can quickly lead to dehydration, which can be very dangerous and kill the child. A little bit about the rotavirus epidemiology. The risk of disease that will bring a child to the hospital or worse is of course smaller, or is of course bigger when the baby is less than 3 months or when the patient is immunocompromised. However, adults and older people will also develop once in a while a gastroenteritis with rotaviruses, definitely when they have often contacts with infectious children. However, the severity of that episode will be, of course, far less than with the smaller children, and definitely when they are premature or immunocompromised. For a long time, people thought that rotaviruses only infected the gut. That is absolutely not true, but that goes for many other viruses. For example, for respiratory syncytial viruses, we think, oh, this is a respiratory pathogen and you can only find it in the lungs. It is not true. When you go and look in the blood during a rotavirus episode, but most people don't do that, of course, but if you would look in the blood, you will find that that virus is viremic. Same with RSV, with an RSV episode. And for when people, for other reasons, do a lumbar puncture in the CSF, you also find the RSV. So we often think that viruses are restricted to one area of the body. Very often that is not the case. And this is also for rotaviruses. When you're in the field, you know that rotaviruses are, or rotavirus infections are seasonal. You can either go in the hospital records and or go in the hospital laboratories and look for the positive isolates or identifications of rotaviruses, and then you find the typical pattern where, and that depends on where you live in the world. But here in Europe, in the winter months or in the final autumn and winter months, you have a peak of rotaviruses, but that will differ from place to place. If you do the same thing, I didn't bring that slide, but if you do the same thing on Google, with Google Trends, you can look for what people look for, and you will find the same seasonality of people looking for the word rotavirus in whatever language online. You can do the same. You can do that for many pathogens. You can do that for influenza. You can do that for many other pathogens, and you will find with Google, you can do Google Epidemiology. It's wildly inaccurate, but it often coincides with the quote-unquote official curves. Every child, almost every child, will develop an episode of rotaviruses. This is something that you cannot escape. At this moment, we do not have evidence of mutations that, and this is unlike for noroviruses, of mutations in the body that, or in a certain receptor that will protect you from rotaviruses. For noroviruses, that's the case. For rotaviruses, we don't have that evidence yet. So everybody will encounter rotaviruses at probably an early age. 1 in 5 children worldwide will develop or will need the medical attention from a physician. 1 in 50 will be hospitalized. 1 in 200 worldwide will die because of a rotavirus infection. When you look at the European region, the first part of the slide is exactly the same. 1 in 1, 1 in 4, 1 in 33, but of course this is very different. 1 in 10,000 will die, and this will lead to, and this is very often in the periphery of Europe, will lead to deaths. And when in Europe an infant dies because of a rotavirus infection, multiple things went wrong. It's not only the rotavirus infection, but it's also a complete lack of medical attention or the seeking of medical attention by the parents that lead to the relatively few fatalities when you compare it with the situation worldwide. But even in Europe, the burden of infection, the burden of disease is definitely before the vaccination era was and is quite extensive. It has gone down. In 2006, about 400 children died of rotavirus infections each year. That has gone down, not only because of vaccination, we'll talk about this in a minute, but also because better access to healthcare, better nutrition, cleaner water, and so on, has led to a decrease, with still some serious hotspots where too many children die because of rotavirus gastroenteritis. The the top 10 countries for rotavirus fatalities are listed here. India, and then definitely Nigeria, Pakistan, and the Democratic Republic of Congo are the most affected countries, or the countries where, besides rotaviruses, they have many other problems that contribute to this situation. All in all, and these are numbers from spring 2016 of the WHO, and based on numbers that were accurate till 2013, 3.4% of the young children that die worldwide die because of a rotavirus infection, with the same sad map, which is so often true for many other infectious diseases, also here being present. One of the things that we do in the research lab in Leuven, where I come from, we study rotavirus diversity. Many people think that rotaviruses are, well, the rotavirus is the rotavirus, yet it is a quite extensive group of viruses with more diversity than meets the eye. That is probably, or that can be explained due to the nature of its genome. Rotaviruses have a segmented genome. Segmented genomes are unusual in human medicine. In fact, there are only 3 virus groups in human medicine that are important and have a segmented genome. And these are, of course, influenza. They have segmented genomes, and there is zoonotic infection. In fact, influenza viruses are avian viruses that learn to infect other species. Another zoonotic virus with a segmented genome is hantavirus. Hantaviruses, primarily in rodents, are also important. And then rotaviruses. Rotaviruses are important pathogens of many animal species. Most of the mammalian species, most of the avian species have their own set of rotaviruses, and because they have segmented genomes, they can exchange these different pieces in their genetic material and make new combinations. The 2 most important ones, or the 2 that contribute to the nomenclature of the rotaviruses, are the VP7 and the VP4, viral protein 4 and viral protein 7, that will designate rotaviruses into G types and P types. It's a little bit like the hemagglutinin and the neuraminidase for influenza. That is true also for the G and P types in rotaviruses. Rotaviruses are complicated viruses in terms of structure. They are multi-layered, and they have a core that consists of VP1, 2, and 3, and inside, of course, is the genetic material. And then they have an intermediate layer here with VP6, which is quite conserved among different rotaviruses. And then on the surface, you have a yellow layer here, and that consists of VP7, and that is accessible to the immune system. And then the spokes of the rotaviruses are the VP4, and they are probably the most interacting with the immune system. The VP7 allows the discrimination of rotaviruses into 33 G types at the moment. The VP4 allows the discrimination of, at this moment, 49 different P types. And every year that number grows. 48 and 49 come from bats, and our nomenclature committee has to designate new ones with fairly strict definitions if most of these types, or all of these types, are at the most 80% on the amino acid level, identical to each other, but there are quite a lot of differences there. When we look at the different combinations of G's and P's, this table only shows you G until 22 and P until 32. In orange, you have the common human strains, that is G1P8, G2P4, that's an exception, G3P8, G4P8, G9P8, and now also G12P8. In green, you have very common animal strains. Here, you have the unusual human strains and the suspected human-animal reassortment isolated either from humans in purple or from animals in blue. And that table is a lot bigger now and is becoming more and more complicated. So because, and that is due to the nature of their segmented genome, they are exchanging genetic information quite readily. Sometimes you have G-types that are emerging. This was true for G9, which was first discovered in the 80s, and then suddenly around 1996, 98, started to become very important. And at one point, in Europe, in many countries, accounted for more than 50% of the rotavirus infections, and then it went down again. A couple of years ago, we were quite interested in the emergence of G12. G12 was first isolated very seldomly, and then suddenly around 2002, it started to emerge. We studied that in a little bit of detail, and it's illustrative for the evolution and the diversity which is hidden in these rotavirus genomes. If you look at prototype strains, which we sequence completely with next-generation sequencing, you have G1 here, or the G1 universe in green, G2 with the DS1 strain in red, and G3 here in yellow. The G12 prototype strain was also known. We sequenced it completely, and we sequenced many others, G12s, and you would think, well, they're all the same. Well, if you sequence the G12, you would think so. If you then sequence from the same viral particle the P, then you see that, well, in the prototype it is associated with P4, which is in fact the P4 which is found in the G2 prototype. And then here you can see that you have G12 associated with P9, P6, P8, which is the most common, and P6, which is in fact the most common in countries such as Bangladesh. And then when you go and look at the other genetic makeup, you see that, well, many of these genes in the G12 prototype strain are derived either from the DS1 or the WA, respectively G2 or G1 prototype. And then when you sequence all the other G12s, you can see, well, they're all G12s, you would call them G12s, but they're extremely different when you look at the complete genetic makeup. In fact, you could say that in the G12s, you have G12s that are actually only G12 because the VP7 is G12, but otherwise they are a G3, or here in red, a G2 prototype, or G12s that are, in fact, except for G12 and or G12 and P6, rather resembling the WA strain. So the diversity behind the G or the P type is quite amazing. And then at a certain point in time, in the beginning of the century, people thought, okay, let's try to vaccinate against rotaviruses. The rationale why people started vaccinating against rotaviruses, except that it was an important pathogen and there was a clear clinical need to do so, was that everybody observed that it's only the first infection with rotaviruses which leads to a severe infection. So a child, when the maternal immunity has disappeared, will have an ever-increasing chance of contracting a rotavirus infection, which will be severe. The first infection with a rotavirus traditionally is severe. When it encounters a second infection, it becomes a serious but moderate infection, and after that, the infections are mild. When you and I encounter a rotavirus, we might have some loose stools that we might not even define as diarrhea, but every year or couple of years, we will be reinfected with rotavirus types. The immunity against rotaviruses is sadly not very long-lasting. So if you can, or if you observe that later infections with rotaviruses are are less severe than the first infection, then it becomes obvious that that's what you want to do. You want to make the first impression that the rotavirus makes on a child a rather more friendly encounter with a live rotavirus vaccine, and that was what happened. And at the moment, two vaccines are on the market. You have ROTARIX from GSK and ROTATEQ from MARC. And these two vaccines are similar enough to be used in similar circumstances, but they're different enough to make things interesting. It's a little bit as with the papillomavirus vaccines that we're currently using. And comparing them is kind of interesting because they are not the same. They do the same things and they work well, but the principle by which they work is different. Well, the principle is the same, that both of them are live vaccines. They're real rotaviruses that you use, but of course they are attenuated. ROTARIX contains, well, only G1P8. It's a monovalent vaccine that uses an attenuated, human-derived G1P8. That was a good choice because at the moment when the people were designing the vaccine, G1P8 was definitely the market leader worldwide, and still is, of rotavirus infection. So that was a good choice. And what ROTARIX does, it counts on cross-protection with other rotavirus types in order to render protection from disease. ROTATEQ has a different philosophy. They started with a bovine rotavirus, which has a machinery which, if you use it in humans, is naturally attenuated. It will not generate overt disease in humans. But in order to humanize this, because you do not want to protect against the G6P5, P5 viruses are not known in human medicine, what they did was they reassorted this with different human G1P8, G2P4, G3P8, and G4P8. And out came these viruses, and they constitute the pentavalent ROTATEQ vaccine. This contains G1, G6, G2, G3, and they're all relevant, but on a P5 background, which is not all that relevant, except for this one, G6P8. Now, the P moiety is the one that sticks out from the particle the most. I would make the case that that P molecule is the most important one to elicit an antibody response, is the most antigenic one, the most important one. So what you see here with ROTATEQ is that P8, and P8 is the most important one in the different viruses, is sort of diluted 1 in 5 in the ROTATEQ vaccine. So if you look at ROTARIX and ROTATEQ, and these are the viruses except G12, I should add G12, but that is also associated with P8 that are in there. You can see that most of the viruses that you're going to protect against are or contain the P8 except G2P4 and G9P6, which is more important in developing countries. ROTARIX has P8 in all of the particles in the live vaccine. ROTATEQ has P8, but it's diluted 1 in 5. That is probably the reason why, for a full vaccination course, you need 2 shots of ROTARIX, but you need 3 shots of ROTATEQ. The reason why you need 3, in my opinion, is the fact that the very useful P8 moiety is a little bit diluted. If I would have to make the vaccine again, I would get rid of the P5 there and upload the vaccine or a second generation vaccine with a little bit more P8. That is the reason why, you can use it in 2 vaccination sessions with ROTARIX. You need 3 at 16 weeks also for ROTATEQ. The reason why you give the rotavirus vaccines so early in life is, of course, because the burden of disease of rotaviruses is early in life. However, the vaccines, both ROTARIX and ROTATEQ, need to be given early and not later on in life, because the risk of intussusception is judged to be too big. Now, intussusception, where the intestine is folding back on itself, was an issue years ago with a predecessor of these vaccines, or the first rotavirus vaccine, that was RotaShield. The RotaShield's program was halted because a number of intussusception cases were seen in industrialized countries more than the background, and thus the program was halted. This was politically not completely correct. This vaccine, even as it was then, the RotaShield vaccine, would have saved probably 10,000 of lives, many more than cases of intussusception that it would generate, but it was taken off the market. The current vaccines, both ROTARIX and ROTATEQ, were evaluated in really big trials. 80,000 plus children, the biggest vaccination trials that were ever done, and it was clearly proven that the intussusception story does not hold up for ROTARIX or ROTATEQ for the current vaccine. So safety is not an issue. We looked at rotavirus vaccination in Belgium, and this was the situation pre-vaccination. Pre-vaccination in our in the laboratory in Leuven, about 20% every year of the gastroenteritis cases were positive for rotaviruses. And then started vaccination, first with the introduction in 2006 of ROTARIX, and then in 2007 ROTATEQ, and the number of cases of rotaviruses went down, and the percentage of positive isolates or positive identifications of rotaviruses stabilized around 6-7%. So it went down from 20 to 6%, and the total numbers went down. What you see here are the number of rotavirus positive cases in blue at my hospital since 1986. What you see in green is the number of births in Belgium per month, and what you see in red is the uptake of the vaccine in 2006. It's interesting to see that the uptake of the vaccine was very rapidly in 2006, and we have about 85-90% of the children that are since then vaccinated against rotaviruses, with about 85% ROTARIX, 15% ROTATEQ. That led to an immediate decrease of rotavirus cases in our university hospital. These were the numbers before ROTARIX introduction, and in the years after that, the numbers of rotavirus cases and rotavirus hospitalizations went down dramatically, not only in the vaccinated children, but also in the unvaccinated siblings that were protected because their brother or sister was vaccinated. When you look at the G types, the G genotypes, you can see that when they started thinking about the vaccination program, here in red, G1 was the most prevalent one. So when they were making the vaccines, they thought, okay, if it works for G1, that's the major target. Then, much to their dismay, just before they launched the vaccine, G1 almost disappeared, and for some reason, either G9 became very prevalent, and G3 was in some years more prevalent, and G4 than G1. But then, luckily for the vaccination companies, just before they launched the vaccine, G1 was there again. But then, after vaccination, when you look at the picture after vaccination, G1 disappeared quite readily, but G2 is now the most prevalent one. This is deceiving, because this puts everything at 100%. This is important for virologists. We love this. And I study diversity. This is nice. But for most of you, this is not important. This is what is left. Yeah? There were a lot of cases of rota in the past. There are few cases now. But of the few cases that are here, yes, a little bit more will be G2P4. We think this is due to the vaccine. We're not completely sure, but this is what we're going with. What is very obvious is that the seasonality of rotaviruses has changed. What you see here in gray is the epidemic curve of rotaviruses between 1986 and 2006. And as you can see, it reaches a peak in January, February. The black line, I'm sorry, but the pointer doesn't work anymore, but the black line is the average rotavirus season. When you look at the colored lines, you can see that after rotavirus vaccines were introduced, the epidemic shifted. It went down, and it was shifted towards the later months. You're coming to help me. Thank you. That's good. Great. Thank you. So as you can see here, the blue line, that was 1 year after vaccine introduction, the orange line, and this is now quite stable. Rotavirus epidemics have all but disappeared from the winter months. It is now a spring infection, but at every point is lower than where it was in the past. So we went completely away from the winter season, and that was good. That was good because now we don't have the perfect storm which happened in the past of influenza, RSV, and Rota at the same time in a pediatric ward. This in fact led to more hospitalizations, and I think rightfully so, of RSV cases that were previously sent back home because people were afraid that when you admitted them in a ward that was already overflowing, they would come in with RSV and then would go out or would stay with either Rota or influenza. This does not happen anymore, and our pediatricians in Belgium can admit RSV cases and or influenza cases more readily than they could in previous decades. So this in fact changed. I mean, the sad situation around Christmas with many people in the hospital changed to children being in Christmas where they should be, and that is at home and not in a hospital bed. I would like to end with, does vaccination immune pressure drive rotavirus evolution? There is some evidence for that. When you look at and that's what we do in the research lab, we make phylogenetic trees with viruses. When you look at the phylogenetic trees based on the complete genomes, or here on the P moieties of the virus, you can see that most of the current strains are in fact lineage 2. However, the vaccines have lineage 3 in ROTATEQ and lineage 1, a lineage 1 virus in ROTARIX. And the distance between the genetic distance between what is in the vaccine and what is circulating is becoming bigger and bigger, and in important regions. When we look at the antigenic regions and we look at ROTATEQ, ROTARIX, the vaccines, and the things that are circulating, which are mainly in green, there are quite a lot of differences between the vaccines in important amino acid positions and what is circulating. When we look at the VP8 structure, you can see that in immunologically important regions, very accessible for antibodies on the VP8, well, there is a difference between, for example, here what is in ROTARIX, a serine, and what is circulating, asparagine, and the same here. In ROTARIX, it's asparagine, in ROTATEQ, and what is currently circulating, it's aspartic acid. And there is a slow but steady move away from what is in the vaccine. The same is true for when you look at the G protein. The current Gs are mainly lineage 1A and 1B, but ROTARIX is in lineage 2 and ROTATEQ is in lineage 3. They're old viruses which are used in the vaccines. Here, the same thing. When you look at the amino acid composition, there are clear differences now between the currently circulating strains and what is in the vaccines. And that is true for most of the antigenic regions, although sometimes the differences are small. Probably here it doesn't really matter if it's an isoleucine or a valine which is circulating, both are aliphatic, but the difference between a methionine and a threonine three-dimensionally is quite large. And the viruses are shifting away, or drifting away rather, from what is in the vaccine. We superimpose this on the three-dimensional structure, and in important positions, immunologically important positions, the number of differences between what is in the vaccine and what is out there is differing. And that's a clear difference. Before we started vaccinating, G1 was predominating. Because of vaccination, G1 has disappeared. The virus, what is in the vaccine, is disappeared almost completely. A lot of the G1 lineages 1 have disappeared too, but some are now surviving, and they are the ones that are causing disease. Now, having said that, these vaccines are still very effective. I mean, we have to do continued surveillance to make sure that the drift between what is circulating and what is in the vaccine does not become too big. But as they are now, they are proving to be very effective. At the moment, these vaccines are introduced in a little bit more than 80 countries now. These numbers are from the beginning of the year, either by their governments or either supported by the Global Alliance, by Gavi. When you look at, and these are part numbers, when you look at different countries and meta-analyses, the reductions in annual hospitalizations for rotavirus gastroenteritis are striking. They are more than 50%. They're typically 75%. They can go as high as almost 90%, and in some cases 95%. So the effect is real, and that is a good thing. And frankly, I do not understand the countries that say, nah, we're not going to vaccinate against rotaviruses. We're not sure yet. That is not really where the evidence lies. The effect is not only on the children that are age eligible for vaccine. This is the reduction in rotavirus hospital admissions, high numbers, but also children not age eligible for the vaccine, the numbers are going down. So the cohort effect is there, and the effect goes beyond the children that are vaccinated. I would like to end with this. This was my son 7 years ago. 134 million children are born each year. 2/3 of them are vaccinated, let's say, decently. 1/3 is not vaccinated. So 33.5 million are non-vaccinated. The cost of a full vaccination course, and that is not including the administration, but just buying the vaccines for a normal standard basic set of vaccines that is used in a standard European country, that costs €32 per child. If you multiply 32 euros by the number that is not vaccinated, you come to about €1 billion. That's it. That's the only thing you need to pay for vaccines for all the children. So in order to do this, you need €1 billion per year. €1 billion, that's a mind-boggling number. So let's compare this with something. The war in Syria costs the US €1 billion per month. You can say, okay, that's far from my bet, I don't care. In Europe, we spend 1 billion every 6 weeks on dog and cat food. Yeah? Now, I will readily agree with you that you cannot starve your dog or cat for 6 weeks in order to pay for vaccines, but still it is food for thought, even literally. We spend on ice cream in Europe 1 billion every 3.5 days. Yeah? So if a Monday, Tuesday, Wednesday, and Thursday afternoon you can go wild again, if that money you would save once a year, then we can pay. So if we don't do this, this is not an issue of is there money enough? The issue is political will, as it is for many things, but this is definitely true also for vaccinations. Thank you very much.